All right, so today we're taking a deep dive into CRT cell therapy. But not just the basics, we're going way deeper, like exploring how single-cell analysis is pushing this already amazing treatment to a whole new level.
It's pretty incredible stuff. It's like we have this powerful weapon against cancer, and now we're getting this super detailed instruction manual on how to use it even better.
Okay, color me intrigued. I've got a stack of research papers here, and they're all buzzing about how single-cell analysis is changing the game. So where do we even begin with all this?
Well, one of the biggest challenges with CRT therapy is relapse. Sometimes those cancer cells find a way to escape the treatment, and they come back even stronger.
Ugh, that's the worst.
Yeah.
So how does single-cell analysis help us outsmart those sneaky cancer cells?
Okay, imagine this. You're trying to track a criminal gang, but you're only looking at their activities as a whole group. You might miss those little differences between individual members.
Right, like you're not seeing the full picture.
Exactly. But what if you could zoom in on each member? You know their unique behavior, their connections.
Uh-huh. You'd get a much clearer picture of how the whole gang operates, maybe even predict their next move.
Exactly. And that's what single-cell analysis does for tumors. It lets us analyze the genomics of each individual cancer cell. It reveals a level of complexity that we just couldn't see before.
So we're talking about reading the DNA of each individual cancer cell.
It is. There was actually this study on diffuse large B-cell lymphoma, DLBCL for short, and they used whole exome sequencing to link a specific mutation in a gene called PTPRA to relapse.
Hold on, PTPRA, now that sounds familiar. Remind me what that is again.
Think of PTPRA like a switch that controls cell growth, and in some DLBCL patients, this switch gets flipped, which makes the cancer cells resistant to CRT cell therapy.
So if doctors could identify this PTPRA mutation, they could potentially predict which patients are at higher risk of relapse, and maybe even change their treatment strategy.
Exactly. That's just one example of how genomics at the single cell level is giving us these critical insights into tumor evolution and treatment resistance.
That's pretty amazing. But it's not just about understanding the enemy right. We also need to make sure our CRT cell soldiers are ready for the fight.
Absolutely. That's where TCR sequencing comes in. It's like giving each CAR T cell a unique fingerprint.
I like that, giving each one a fingerprint. So what are we learning about these cells by tracking them like this?
Well, one surprising thing is that after we infuse the cells into the patient, the diversity of the CRT cells actually decreases. It's like a few elite soldiers end up doing most of the work.
While the others kind of fade away.
Yeah,
So if we could figure out what makes those elite clones so effective, we could potentially select for them before treatment.
precisely. That's one of the most exciting things about TCR sequencing.
Wow. So we're identifying the best targets on the cancer cells, and then we're actually optimizing the CRT cells themselves. What other roadblocks are we tackling with this single-cell analysis?
Well, a big one is T cell exhaustion. It's like those awesome cancer-fighting cells get tired and they just lose their effectiveness over time.
Yeah, like they hit a wall. So how is single-cell analysis helping us figure out this exhaustion?
This is where epigenetics comes in. It's not about changing the DNA sequence itself, but understanding how genes are turned on or off in those exhausted CAR T cells.
So we're looking for the off switch, the one that's making them burn out.
Exactly. And a technique called SCATAC-SEC is allowing us to do just that.
Okay, SCATAC-SEC. Break that down for me. What is it measuring?
Imagine the DNA in a cell is like a long, winding road with lots of gates along the way. SCATAC-SEC tells us which gates are open.
So in an exhausted CAR T cell, we'd see certain gates are shut, preventing them from getting to the genes they need.
Exactly. And even more exciting is that researchers have used SCATAC-SEC to identify specific transcription factors that are responsible for closing those gates.
Transcription factors. Remind me again what those are.
They're like master controllers that regulate gene expression. They can bind to DNA and either activate or repress the transcription of nearby genes.
Ah, so they're like the key masters. They control which genes are turned on or off.
I like that. And by understanding which transcription factors are involved in T-cell exhaustion, we can start developing drugs to target them, potentially preventing or even reversing the exhaustion.
Okay, so we're getting a handle on the cancer cells. We're making sure our T cells are in fighting shape.
Yeah.
And now we're even figuring out how to keep them energized for the long haul. This is amazing.
It is. And there's a whole other level of information we can get from single-cell analysis. Transcriptomics.
Okay, I'm ready for that. But maybe we should take a break here and pick up with transcriptomics in part 2.
Sounds good to me. So last time we left off with transcriptomics.
Right. It's like we went from the blueprints to actually seeing which ones are being used. What exactly are we looking at here?
Well, think of it this way. If genomics is the parts list for a cell, transcriptomics tells us which parts are actually being put together.
Okay, that makes sense. So we're seeing what the cell's actually doing.
Exactly. And the technique we use to analyze this at the single-cell level, it's called RNA-seq, or single-cell RNA sequencing.
And what kind of insights are we getting from this when it comes to CRT cells?
Well, for one, it's helping us categorize CRT cells into different types based on their gene expression profiles.
So we can tell which ones are the attackers and which ones are the support crew, just by looking at their RNA.
That's right. And this can be really valuable for predicting how well a Kia T-cell product will work.
Okay, that makes sense. But is cRNA-seq helping us figure out how to actually make these therapies more effective?
Absolutely. Remember how we talked about the potential dangers of too many CERR genes integrating into a T-cell?
Yeah, it's like the CRT cells were getting overloaded.
Exactly. Well, cRNA-seq is helping us fine-tune that process.
So how?
Researchers are using it to track the expression levels of the CRR gene in individual cells. This lets them see how different gene editing techniques and manufacturing processes impact CRR expression.
And ultimately, how well the T cell functions.
Right. It's all about finding that sweet spot.
Enough CAR expression to kill the cancer cells,
Yeah,
but not so much that the cell burns out.
exactly.
That's amazing.
And here's another thing. We can use cRNA-seq to predict on-target off-tumor toxicity.
Right, that's when the CRT cells might attack healthy tissue.
Exactly. But imagine being able to analyze the gene expression profiles of the tumor cells and the healthy cells in a patient.
And if there's too much overlap, we know there's a risk of those side effects.
Right. And doctors can then adjust the CRT cell therapy or even consider other treatment options.
That's incredible.
And we've just scratched the surface of cRNA-seq. There's so much more we can learn about CRT cell behavior, the tumor microenvironment, and how to really personalize these therapies.
So we've talked about individual cells, but what about the bigger picture? The tumor microenvironment.
You're right. That's where the real action happens.
So how does single-cell analysis fit into that?
Well, one approach is to analyze the types of cells in the tumor microenvironment using single-cell techniques.
So we're identifying all the players in the game.
Exactly. And this can tell us a lot about how the tumor microenvironment might affect CRT cell therapy. If we see a lot of immunosuppressive cells, you know, the ones that suppress the immune response, we might expect the CRT cells to have a harder time.
So we could adjust our strategy accordingly.
Right.
But analyzing the types of cells is only part of the story, right? We also need to understand how they're interacting with each other.
You got it. And this is where cRNA-seq comes in again. By analyzing gene expression patterns, researchers are starting to figure out the communication networks between cells in the tumor microenvironment.
So we're eavesdropping on their conversations.
Exactly. And these conversations can reveal how the tumor is affecting CRT cell function.
So some tumor cells might be sending out signals that suppress immune activity.
Exactly.
So by understanding these signals, we could potentially develop drugs to block them.
That's one possibility. Or we could engineer CRT cells to be resistant to them.
So we're really taking control of this battle.
It's truly revolutionary.
So we've gone from analyzing individual cells to understanding how they interact in the tumor microenvironment. And we've seen how single-cell analysis is changing how we make CAR-T cells. What about the manufacturing process itself? Can this technology help us improve that as well?
Absolutely. Remember, CAR T-cell therapy involves taking a patient's T cells, engineering them to express the CAR-T, and then multiplying them before putting them back into the patient.
Right.
Well, traditional methods for assessing CAR T-cell products were like doing a random check on a factory production line.
But with single-cell analysis, it's like having a camera on every single worker.
Exactly. We can now analyze each individual T cell, making sure it's been engineered correctly, that it's expressing the CAR at the right level, and that it's not showing signs of exhaustion.
That's amazing.
And it's not just about quality control, it's about actually optimizing the engineering process.
How so?
Well, remember our discussion about CAR integration and how too many copies of the CR gene can lead to exhaustion.
Just like the cells were getting overloaded.
Right. Well, single-cell analysis is helping us fine-tune the gene editing techniques so we can get the optimal level of CAR expression without causing exhaustion.
We're giving those CAR-T cells the tools they need without overwhelming them.
Exactly.
This is incredible.
And it's all about making CAR T-cell therapy as precise and personalized as possible. And there's another area where single-cell analysis is making a difference: spatial transcriptomics.
Spatial transcriptomics, what's that all about?
Well, cRNA-seq gives us great information about gene expression, but it doesn't tell us where those cells are located in the tumor microenvironment.
So it's like knowing the ingredients, but not the recipe.
Exactly. Spatial transcriptomics adds that spatial dimension.
So how does it work?
There are different techniques, but basically it involves capturing RNA transcripts from specific locations in a tissue sample and then sequencing them.
So we can see where those cells are located within the tumor.
Exactly. It's like creating a 3D map of gene expression.
That's amazing.
And researchers are starting to use it to study CRT cell therapy.
So what are they finding?
Well, one study looked at how CRT cells get into solid tumors.
That's one of the biggest challenges, right?
It is. And this study showed that CRT cells tend to gather around blood vessels.
Makes sense. They need a way to get in and out.
Right. And another interesting thing, they also found that CRT cells were often near areas of high immune activity.
Like they're drawn to the action.
Exactly.
So spatial transcriptomics is showing us where the CRT cells are. And that could give us clues about how to improve their infiltration.
Exactly.
This is all really mind-blowing.
It is. And as spatial transcriptomics gets better, it will have a huge impact on how we design and deliver CRT cell therapies.
So we've gone from analyzing single cells to understanding how they interact in the tumor microenvironment. And we've even seen how single-cell analysis is changing how we manufacture CRT cells.
It's an incredible time for this field. But it's important to remember that single-cell analysis is still pretty new. There are definitely challenges that researchers are working on.
Okay, so what are some of those challenges?
Well, one problem is that it can be tough to separate cells from tissues without damaging them, especially with solid tumors.
Yeah, they can be pretty tough to break apart.
Exactly. And any damage can affect the results. Another challenge is all the data.
Yeah, we talked about those massive data sets.
Right. And making sense of all that data requires some pretty complex tools.
So we need better ways to analyze and understand all this information.
Exactly. But the good news is that the technology is getting better all the time.
That's good to hear.
It's an exciting time, and I think we've only just begun to see what single-cell analysis can do.
Okay, so we're back, ready to wrap up our deep dive into single-cell analysis and CRT cell therapy.
It's been an amazing journey. We've covered so much ground.
We have, from individual cells
Yeah.
to the tumor microenvironment, and how single-cell analysis is changing the way we treat cancer.
And there's still so much more to explore.
Okay, so what's next? What are the big challenges and opportunities on the horizon?
Well, one of the biggest hurdles is scaling this up, for clinical use.
Right now, it's mostly happening in research labs.
Exactly.
Yeah.
But scientists are working on making it more practical, more streamlined, and more affordable.
So that more patients can benefit from it.
Precisely. Imagine every cancer patient getting treatment that's tailored to their specific tumor.
That would be incredible.
Another exciting area is the development of new computational tools.
Right. Those massive data sets we talked about.
Exactly. We need better ways to analyze all that information.
To find the hidden patterns.
And luckily, the field of bioinformatics is exploding right now.
So we're getting better at deciphering those secrets.
It's like having a whole team of code brushers on the case.
I like that analogy.
Yeah.
So we've got the technology evolving and the data analysis tools catching up.
It's a perfect storm.
And it feels like we're on the verge of a major breakthrough.
I think so too.
This has been an incredible deep dive.
It really has.
We've learned so much about the power of single-cell analysis and its potential to revolutionize CAR-T cell therapy.
And it's all thanks to the tireless work of scientists all over the world.
To all those researchers pushing the boundaries, we salute you.
And to our listeners, we hope this deep dive has inspired you to learn more about this amazing field.
Because the world of single-cell analysis is constantly evolving, who knows what incredible discoveries are just around the corner.
That's the beauty of science. There's always something new to learn.
This has been The Deep Dive. Thanks for joining us.
Until next time, keep exploring.